Okay, so you go to this twistbioscience.com and so you can make an account. I think you need to give the lab, maybe your RICE email might be better. Okay, so if you recall, you created this small region, right? So this is around 170 bases. And so the only constraint is Twist has a size limit on how big of a fragments they would let us order. So their minimum is 300 bases and the maximum is 1800. So the reason why I was ordering together with other things, 5 KB. Okay. If you want the G fragment is 1.8. Okay. Yeah. So when we ordered this gene fragment, this size limit is between these two ranges. But if you order something called clonal fragment, what they do is not just give you the double-stranded DNA, but they'll put it in a larger plasmid for you. So that way you can put it into bacteria and make more of it. But we ordered this because it's quicker. For the other one, they take 5 extra days. Yeah, so if you're doing a very small region, like what we are doing right now, they won't let us order this because they cannot manufacture this very easily. Maybe it's not cost effective for them, right? So one thing we would do in that case is just make it bigger. It doesn't matter how much extra fragment we get. So just make it until it reaches 300 bases.
Yeah, 300.
So we could either do that, or if you have someone else in the lab wants something else, right? So you can put your fragment and their fragment next to each other. And if they're both meant for standards for PCR or ddPCR, you can both use the same thing independently. It doesn't matter, right? So that's one thing I'm doing right now, where we are fusing multiple fragments and then ordering. So you can save a lot of money, both shipping and manufacturing this.
Yeah.
Okay, so once we sign in, so there are multiple categories here, and the one we are looking at is specifically the one that says genes. It will ask you for a name of the project. We'll just enter any name, and so we'll paste the sequences in here. Yeah, so it gives us 2 options again. So the first one is where it just gives you the linear fragments, and this is the one we prefer for the standards for both qPCR and ddPCR. For ddPCR, I guess it doesn't matter as much. So this is cheaper. It's 7 cents per nucleotide. The other one is where it puts the thing into a larger plasmid, and that is slightly more expensive. Not that it matters much, but you can order longer fragments. If you're ordering a larger things, you might want to do this. And if you want only linear DNA from this, you can amplify from here. Yeah, so also the turnaround time is around a week for this, and this one is 2 weeks or more. So we'll click on the gene fragments, order now. So we are selecting nucleotide, and we will manually paste the sequences. So I'm choosing the right side part. If you have 10 or more, you might want to make them into a file separately, a CSV file, and then upload them using the left option. Currently, we have only 2. So I'll go here and I'll name. Okay, so assuming we selected this 300 base thing, I'll copy it. Okay, so I'll select from here to here. I'll copy it and we just paste it there, and we'll name it RSV consensus standard. And the second region we were supposed to order is where we copied this region, but we mutated it slightly. And the 2 mutations I made are indicated here.
Mm-hmm.
So the one more thing in benchling is when you copy over anything, you want to make sure you make changes in it. That way, someone in the future can refer to it and see what has changed here. So I made this mutation G2A, and I also probably wrote in the comments section here. Yeah. So you see, I made these 2 mutations in the sequence file to accommodate the probe from this paper. So that way, anytime someone opens the sequence, they can access that paper. They know exactly where we got the probe and primers from.
Okay, yeah. That makes
Yeah.
sense.
So again, we have this smaller region here, 170. Since this is too small, we'll just make it bigger. Maybe just select this whole thing. It's 306. So you copy this, you paste it here, and you name it RSV probe binding. Yeah, so we just copied the sequences here, named them. Now we'll say continue. And we'll stop just before we order because we don't want to order right now. So you can see the cost of each of these. It depends on how long they are. So this one is slightly shorter and this one is slightly longer. So the cost is appropriately scaled. Also, they charge for shipping. I think they charge some $20 for shipping. But I emailed this company a while ago and they said whenever you, right before you place the order, before you continue and make the payment, if you email them with this quote number that this generates, what they will do is they'll waive the shipping charges for us. So I specifically sent them from Silberg Lab, but maybe they don't care even if it's from Stadler Lab, so it should work.
Mm-hmm.
So then you just press continue and then you place the order and it will
That's
go
cool.
through. Yeah, so there are options here. We just select the default, which is 2 ml tube. If you have multiple, if you have like 50 things you're ordering, you can get them sent in a PCR plate.
Mm-hmm.
And there are 2 options here. Either of them, it doesn't matter which one you select. But if you cap it at 1000 nanograms, that means you're getting a defined mass of the product. And so if you recall, when we get this tube, it will have the powder, just like the primers. And to that, you will add a specific amount of water. So for example, if they're sending, so I think for gblocks, what I've been doing is I just add a defined volume of water. Just say, let's just say 200 microliters. No matter if I get a 1000 nanogram or if I just say send everything, that way we'll get slightly more DNA, although we don't require as much DNA. So you just add 200 microliters. So the concentration in this case would be 1000 divided by 200. So that would be 50 nanograms per microliter, right?
Mm-hmm.
And so if you want to calculate the mass of this, you go to, let's say, NEB's calculator DNA mass. So if you Google that, then the first thing that shows up is the double-stranded DNA mass to moles converter. So here we have the amount of mass in the product, but we don't know how many molecules there are. So if you have an approximate length of the DNA, it will approximate that length to a given number of mass per nucleotide, and then it will calculate how many molecules there are. If you want it to be fully specific, you can give it the full sequence. That way it knows exactly what the mass of the A, T, G, and C. But if you don't, it will just average the mass of all 4 of those and take that as the mass per nucleotide. So you can just give it the length. So one of our things had 317 bases. And the mass that we're getting is 15 nanograms per microliter, right? So we'll just say 50 nanograms is the mass, and it will tell us how many molecules of these there are. So it has 255 moles of DNA, femtomoles, and it converts that using Avogadro number, and it's giving you the copy number. So this is 15 nanograms per microliter. You have 1.5 e to the 11 copies per microliter.
Okay.
So when we do ddPCR, if you remember our calculation,
Mhm.
we need 50,000 or we need a maximum of 5,000 copies.
Yeah.
That would be 5,000 for 10 microliters that goes in. So according to that, you need 500 copies per microliter. So, and if you take the vial that we get, we suspended it in 200 microliters, that is giving you
Mhm.
this mass, and you have to dilute it all the way from 1E11 to 500 copies. So you dilute it like 7 orders of magnitude.
Yeah.
So that's how we do the calculation of how much to dilute. So we dilute 100-fold and 100-fold again and 100-fold again. So
I see.
that's the whole reasoning for this whole thing. But so the ordering part is just straightforward. So now that you said this, you can just cap it and 2 ml tube. And once you say request code, it will generate a code and it will add the shipping charges to it. And I can forward you the email of that person that you have to email in case you're ordering in the future, and then they can waive the shipping charges for you if you want.
Okay. I guess just my rice.
Yeah.
Yeah.
Yeah, I think that's about it. Do you have any other questions?
No, I don't think so. Not at the moment.
Yeah, so I'll save this video somewhere where people can find it. So in case you want Lauren or anyone to order, you can help them out the first time, and the future they can order. But technically we need just to order once this gblocks, and it will last a whole lifetime because we're diluting seven orders of magnitude.
Yeah. Can you show me or help me with the calculations for the dilutions?
Yeah, I can do that once it comes
Okay.
in.
Cool. I might go and do that this evening. So.
For the primers and probe? For the primers?
No, for the influenza standards.
Oh, do we have new standards or are these the same old ones?
I think it's the same old ones. Or do you remember if we finished it last time we did
Oh,
dilution?
yeah. They were left outside, right?
Possibly.
Oh, that's something else. Okay. Yeah, so if you're starting with this vial with the green cap that comes from Twist, we can check how much mass it has and how much we resuspend it in. So usually when I resuspend it, I try to write the volume on the side, or I write the amount of nanograms per microliter it has been resuspended to. So that way in the future, if you come back after 6 months and open the same old vial, you know what is the mass per volume.
Mm-hmm. Okay.
So we can just start from that and calculate.
Cool. Sounds good.
Yeah, so